Hello everyone, my name is Cindy Lopez and today I am going to be giving you guys an overview on the different phases of the cell cycle. The cell cycle is broken up into two main phases, the M or mitotic phase and interphase. M phase consists of many different subphases that I will get into later, and as well as cytokinesis. Interphase is broken up into three subphases known as G1, S, and G2 phases. More details later. Let's begin with M phase, the nucleic and cell division of cells. As mentioned before, the M phase is broken up into five subphases: prophase, prometaphase, metaphase, anaphase, and telophase. In this slide, I've provided a quick chart on how the flow of the M phase works from the beginning to the end. Here I'll be giving a little more details on prophase. It is the initial phase of mitosis. This is where the nuclear envelope breaks apart, or begins to break apart, I should say, and allows for the free chromatin to begin to organize. Chromosome condensation occurs here, and this is when the free-floating chromatin in the nucleus becomes compact and fold into each other, becoming more and more organized. Once the chromatin has become organized, the organized structure is now called chromatid, and two chromatids come together, joined by the centromere. At the same time, in this phase, centrosomes, which are microtubule organizing centers, have microtubules that begin to assemble the meiotic spindles. Once the organized chromosome can be seen via microscope, a cell is considered to be in prophase. Here I provided an image where you can see the free-floating chromatin in the nucleus as it is about to break apart. In prometaphase, the nuclear envelope is fully broken down, and you can have chromosomes begin to become more organized. The spindle poles are at opposite ends of each other, and the spindles start to grab onto the chromosome at the centromere through the kinetochore. Here we can clearly see the spindle poles, which are in green, and how the spindles themselves are reaching out to grab onto the chromosome. It's kind of cool. In metaphase, chromosomes are aligned at the equator of the cell, forming a metaphase plate. The chromosomes do appear as if they are stationary, but they are actually being tugged apart by the spindles. There is a sort of tug-of-war going on where each end is trying to get one of the sister chromatids to come to its side. Here you can see how the chromosomes are organized in the middle of the cell, and the spindles are trying to grab them. In anaphase, the chromatids are separated. They are being pulled to their designated spindle pole, and anaphase is also known as the shortest phase of the cell cycle. So these chromatids are being pulled at a pretty quick rate. I should mention that there are two types of anaphase A and B. In anaphase A, the chromatids are moved because the microtubules are becoming shorter. In metaphase B, the movement of the spindle poles occur because the microtubules are becoming elongated. Here we see that the chromatids are no longer together and are being pulled apart towards their designated pole. Finally, we've reached telophase, which is the final stage of mitosis. The chromosomes uncoil into extended fibers. The daughter chromosomes are at the opposite poles completely, and at this point the spindles disassemble and the nuclear envelope starts to form again, encapsulating the chromosomes. Finally, the very last step is cytokinesis, which is seen by the formation of the cleavage furrow and the full separation of the two daughter cells. Here we can see that cleavage furrow forming in the middle of these cells, indicating that they're at the end of the M phase. Something I should mention here is that although a lot happens in the M phase, the overall process takes a relatively short time compared to interphase. We can see that here clearly in this figure. Interphase is defined as the growth phase of eukaryotic cell cycle. It has three subphases itself, the G1, S, and G2 phase. The G1 phase can vary greatly in time, but typically this is when protein and organelles needed for DNA synthesis are duplicated. In S phase, DNA synthesis occurs in the nucleus of the cell, and then finally in G2, chromosomal DNA replication is completed, and the whole cell cycle begins again as the cell makes its way to the M phase. Thank you so much for listening to my presentation, and here are my references.